Hi there. This video is on organelles, the origin of organelles, and some of the organelles that are in different cells. And I'm talking about membrane-bound organelles, which is what I officially call an organelle. So I'll start talking generally about organelles, how organelles are formed, the evolution of those, and then talk specifically about mitochondria. In part two, I'll talk mainly about chloroplasts, but also some other organelles as well. The idea that organelles such as mitochondria, chloroplasts, and others originated from an endosymbiotic event, also called symbiogenesis, was sort of rediscovered or redescribed by Lynn Margulis in 1967. Some of the original ideas came from Konstantin Mershkowski from Russia, and that was back in 1905 to 1910. But it was hotly debated, and so it wasn't until Lynn Margulis came out with her paper that it sort of rekindled the discussion of the origin of these organelles. And in fact, it took a while. Lynn Margulis sent her paper to 15 different journals and was turned down by the initial 14 before it was finally published in the Journal of Theoretical Biology in 1966, or first submitted in 1966, and it was published in 1967. You'll notice that it's under Sagan, Lynn Sagan. She was married to Carl Sagan, the astrophysicist at the time. Even after the paper was published, and she published other papers on the same topic, she had to really be persistent about her ideas, and finally, after a decade or so, it really caught on, and now with molecular techniques, it's clear that the mitochondria, the plastids or chloroplasts, and other organelles originated as endosymbiotic events. So here's the general scheme of how this occurred. You have a bacterium up here with its own chromosome and a free-living bacterium, and then a free-living protist, unicellular with a nucleus. Often there's a time when the two of these actually are mutualists. They live together and provide things to each other. So there's a time when they actually habitualize to one another and get used to communicating and sharing resources back and forth. But then at some time, one of these organisms enters the host cell and becomes either a parasite or a mutualist at that time. And some of the DNA actually from that moves into the nucleus and is expressed from the nucleus. So that genome in the parasite or mutualist, eventually the organelle, becomes reduced, and the genome of the host increases somewhat over time. Also, the mutualist here is gaining some nutrients from the host cell as it lives in there. Eventually, a lot of these genes that have been moved to the nucleus of the host are expressed from that nucleus, and those proteins which have to function in that now organelle have to be transported through the membranes and then become functional proteins in those organelles. So this is how the mitochondria became incorporated into eukaryotic cells. Eukaryotic cells already had a nucleus. You get this alpha proteobacterium entering, and that becomes an animal or a protist cell. Plant cells were already cells with mitochondria. That event happened earlier. And then later they took on a cyanobacterial ancestor that became the chloroplast. Over the next several million years, DNA from the organelle, the mitochondrion, moved into the nucleus. Now, some of that became functional as genes in the nucleus, so then they were transcribed in the nucleus. The RNA moved out into the cytosol. That became translated in the cytosol and then moved into the mitochondrion as proteins that were functional in the mitochondrion. Similar things have happened with animal or plant cells, where some of the DNA, much of it, has moved into the nucleus from both the mitochondrion and the chloroplast. There is some evidence of some of the genes also moving from the chloroplast into the mitochondrion. And I think I saw one report of a gene from a mitochondrion actually moving into the chloroplast and being expressed from there. Additionally, there are protist cells that have picked up a photosynthetic eukaryotic cell, and then that has been incorporated into those protist cells as something that's sort of like a chloroplast, but it actually starts out having a chloroplast, a mitochondrion, and a nucleus. And those genes from the chloroplast, from the mitochondrion, move both to this nucleus and to the main nucleus in the host cell. And then mitochondrial genes also have moved from the host cell, from the mitochondrion of the host cell into the nucleus. So the nuclei of these tend to have a combination of genes from many, many different sources. You can also have genes transferred from bacteria, from archaea, through viruses, etc. So that by the time you look into the nuclear genomes of all of these, they have a mixed ancestry as far as the genes that are included in those genomes. This is a graph of genome sizes down here, base pairs per genome, nucleotides for viruses, RNA viruses. So here's 1,000, there's 1,000,000, there's 1,000,000,000, etc. And everything up here in black, those are free-living organisms. The ones in kind of this brown color or olive color are a mixture of parasites, organelles, and free-living organisms. And then everything in white here, those are all obligate parasites and organelles. So the tendency for bacteria is once the bacterium becomes a parasite or an endosymbiont or an organelle, it will tend to lose DNA and be down here. The same with some eukaryotes that have become parasites and some have become organelles. So the trend is for the genomes to be reduced in those. And one of the main reasons for that is they're getting a lot of their nutrition and molecules that they need from the host organisms, so they don't have to make them. Those genes can be free to mutate that are normally used for those functions and then eventually disappear. So this is a phylogram showing some of those events. So the bacteria are up here, and then the archaea are here, and the eukaryotes are here. So the first endosymbiotic event that we know about, although there were probably more than this, are these planktomycetes coming over here into this proto-eukaryote, becoming the eukaryote with a nucleus. And then that happened about 2.4 billion years ago, about 2.2 billion years ago, then an alpha proteobacterium, similar to a modern-day rickettsia, which are parasites, moved into those cells and became mitochondria because they could do oxidative phosphorylation. So that was an advantage in an atmosphere that was becoming more oxidative. That is, it had more oxygen, which was toxic to organisms that were anaerobic and couldn't deal with that oxygen. And then these events, this event, at least one, probably two, where a cyanobacterium moved into one of these protist eukaryote cells, and those became the plastids. There are other arrows down here that you can't see too well, but these are red algae and green algae, that is eukaryotes, moving into other protist cells, other eukaryotes. So it's eukaryotes inside of eukaryotes. and one of those is becoming the organelle, and the other one is the host cell. If you refer to a timeline, this being 4.55 billion years here, and this is modern day, then these endosymbiotic events that led to eukaryotes and eukaryotes that could live in oxygen, that is aerobic eukaryotes, happened about 2.4 to 2.2 billion years ago, which coincides with a time when oxygen levels in the atmosphere and in the water and the oceans was increasing. So it would be an advantage for a eukaryote that was normally anaerobic to become aerobic by picking up one of these bacteria that could do oxidative phosphorylation, essentially protecting that cell from the oxygen, but oh, by the way, making much more ATP, so it was a double advantage. Then about 1.9 billion years ago, there's this initial endosymbiotic event of a cyanobacterium going into a eukaryotic cell to become plants. And then there are other things happening down here, but there's also another endosymbiotic event creating something like a chloroplast in the group called Rhizaria as well. So there are at least two events where cyanobacteria were picked up, and those became organelles that accomplished photosynthesis in those organisms. Because of these endosymbiotic events leading to organelles and different organisms then, our view of evolution has changed a bit. So that if you have this first eukaryotic common ancestor, and then you get this split between bacteria, archaea, and eukaryotes, and then with a mitochondrion entering that alpha proteobacterium, entering this eukaryotic cell to become the mitochondrion, that's what people had previously pictured, or the concept of this evolution had been. But now we're thinking that this is more like it. This is the last universal common ancestor, and then you get a split between bacteria and archaea. But then to form eukaryotes, you don't come out of that same line. You actually join together two or more, usually, bacteria with an archaea to get eukaryotes. So there's a difference in how these organisms come together to produce a eukaryote. So now I want to concentrate on some of these initial endosymbiotic events to form eukaryotes. And so the first one here is a planktomycete, or a planktomycete relative. These have internal membranes. So for bacteria, they're kind of unusual in that they have internal membranes. Some of the membranes, some of the organelles actually look like nuclei, so that's why it's thought that these were the source of a nucleus in this eukaryotic cell. So it was a planktomycete with an archaeal relative. And then an alpha proteobacterium entered that to become the mitochondria. And this occurred about 2.2 to 2.4 billion years ago. So here it's depicted in a few pictures to get the concept that the progenote cell was an archaea, and then the planktomycete, which had internal membranes, joined with that to become the initial eukaryotes, which would have the host cell and then internal membranes, including a nucleus. Next, a purple bacterium, alpha proteobacterium, then joined with this eukaryote with a true nucleus to become the organelle that produces oxidative phosphorylation, that is, takes in oxygen and produces a lot of ATP, and that is the mitochondrion. Here is an artist's depiction of a mitochondrion with the outer membrane shown out here, and then the inner membrane, and then this inner section is the matrix, essentially the cytosol of the mitochondrion. And if you look down here, a little tiny picture, so I'll show a better one in the next one, but there's the outer membrane, the inner membrane, that cytosolic portion, And then there's a space between the inner and the outer membrane. Here is a more detailed view where you have the outer membrane here. There's the inner membrane, and you see it's folded inward so that you can have more membrane. And on that membrane are the protein and proteins complexes that accomplish oxidative phosphorylation. So that's why the inner folding of that inner membrane. And inside there is the cytosol of the mitochondrion, which is known as the matrix. And then there's an intermembrane space between the two membranes. And of course, this organelle has DNA, so there's the chromosome in there. And each one of these little balls that you can see here, often people call them lollipops, that's the ATP synthase. So it has an F0 portion, which sticks through the membrane, all the way through to from the intermembrane space through to the matrix. And then there's a head portion called the F1 portion, and that contains the ATP synthase function of that multi-protein complex. So some textbooks will tell you that the ATP synthase has flipped from one side to the other during evolution, and this is actually not true. It's pointed in exactly the same orientation as it was all the way back to the oxidative bacteria where it formed first. So here's a gram-positive bacterium, and the ATP synthases are pointing into the cytosol of that. So they acidify the outside of the cell by pumping protons out there and allow them to flow back in to produce the ATP the ATP. In gram-negative bacteria, they have two membranes, and you have the heads of the ATP synthase, again, sticking into the cytosol. They acidify an area between the two membranes, and then allow those protons to move through the ATP synthase to produce ATP. When you look at the membranes in mitochondria, the difference is that inner membrane is folded in and out within that central space called the matrix, which is the cytosolic portion of the mitochondrion. And you see the ATP synthases again are pointed in towards that cytosolic part of the mitochondrion, and that's where the ATP is produced. And then finally, in chloroplasts, what you have are the ATP synthases are on the thylakoid membrane, and those membranes are formed by an infolding again of that inner membrane of the chloroplast. And then the ATP synthases again are pointed from the middle of the thylakoid out towards the stroma of the chloroplast, which is analogous to the cytosol of a bacterium. So they have not flipped at all. They're exactly in the same orientation that they have been for billions of years. So back to the mitochondrion, you have pyruvate, also fatty acids, and amino acids, which can be converted into pyruvate and or acetyl-CoA. And within the matrix of the mitochondrion, you have the TCA cycle, producing CO2 along with a lot of NADH and electrons. Those move into complex 2 and into complex 1 and complex 2. The electrons move through coenzyme Q and then through complex 3 and then cytochrome. Then in complex 4, you have oxygen combining with water and protons and electrons. And finally, those protons are pumped through complex 5, which is the ATP synthase, to make ATP. And all of these complexes here are on the mitochondrial inner membrane, including complex 5, whereas the TCA cycle is just within the matrix. It's not on a membrane structure. With the exception of this reaction, succinate to fumarate, which is actually part of the complex 2 within the membrane. So here's that process, starting with NADH here, which is produced in the TCA cycle. NADH is then oxidized to NAD+. The protons go through that structure to the inner membrane space, and the electrons move down the chain to first complex 2, which is that fumarate to succinate compound, or succinate to fumarate. And then the electron moves down further, you pump more protons through to the inner membrane space, and then finally complex 4 takes the oxygen, such as that you breathe in, combines it with protons, electrons to become water. That is a energy producing reaction, so some more protons are pumped over through to the intermembrane space. And then all those protons flow back through the ATP synthase via a concentration gradient called the proton motive force. And ADP plus inorganic phosphate is converted to ATP. So many of these reactions are redox reactions. So in this case you're oxidizing NADH while at the same time reducing the flavoprotein, and then you're using that to oxidize a different compound, and on and on. So these are just a series of redox reactions finally to produce water from oxygen, protons, and electrons, and moving those electrons down, which lose energy all the way down, and that loss in energy is transferred to pumping protons across the membrane through those complexes, and then eventually those protons going with a proton concentration gradient through the ATP synthase, and then producing ATP on the matrix side of that mitochondrial membrane. So when and why did this alpha proteobacterium become so beneficial to those eukaryotic organisms? So we want to look at when that happened and why it happened. And this diagram actually answers both of those questions. The why is that at that time cyanobacteria became oxygenic. So before that there were cyanobacteria here back to about 3.5 billion years ago, maybe 3.8 billion years ago. But they did not give off oxygen. But once they got that manganese center that incorporated into photosystem 2, then they became oxygenic. So they actually could get more energy that way. But they gave off oxygen, which was a toxin to most organisms. And so a lot of organisms began dying, and unless they could pick up a way to get rid of the oxygen, sequester it, somehow protect the cell from that oxygen, then they wound up going extinct. So that worldwide oxygenation event began somewhere between 2.5 and 2.7 billion years ago. And by about 2.4 to 2.2 billion years ago, you either had to go deep in the ocean or deep in the water to avoid the oxygen, or somehow become used to living in the oxygen by being able to detoxify it. So early on Earth, and all the organisms actually at this time were still living in the water. There were no terrestrial organisms. That didn't occur until probably about 1 billion years ago, when there finally was a stable magnetic field, which then diverted different cosmic rays, and also an ozone layer had to build up also to protect from the UV irradiation that was coming. Both of those would just make anything on land sterile. It would sterilize the area because it would destroy the DNA and the proteins in those cells. So early on, again, you had low oxygen in the atmosphere, low oxygen in the water, all the way to the bottom. And so for the anaerobes living at that time, and everything was anaerobic, or nearly everything was anaerobic by that time, it worked out well. So bacteria that grew in low oxygen could live up high in the water column, low in the water column. The eukaryotic cells could also, and these are without mitochondria at this point, could live in low oxygen, either high or low in the water column. So about 2.5 to 2.7 billion years ago, oxygen levels began to rise. And by the time you get to 2.2 to 2.4 billion years ago, it became pretty drastic. A lot of organisms were dying because the oxygen essentially oxidized the proteins and the DNAs in their cells, and they died. Because bacteria were pretty adept at mutating and adapting to new environments, then some bacteria began to sequester the oxygen by attaching it to certain molecules or taking it through certain reactions that would protect the cell from that oxidative stress. One of the reactions was taking oxygen, joining it with hydrogen to make water, which is just fine. And that was the basis for oxidative phosphorylation. So the original purpose of that reaction, oxygen plus hydrogen to make water, may not have been to produce ATP, which it ultimately did, but was mainly to sequester the water, or to sequester the oxygen, and to produce water. So that way the cells could be protected and produce a useful compound in the process. But eventually the bacteria did develop a system which we call oxidative phosphorylation for using the energy from that reaction. Oxygen plus hydrogen yields a great deal of energy. And they use that energy to create a electron transport chain, which in the process pumped protons across a concentration gradient into the intermembrane space. And then the ATPases, which were already there, the ATP synthases, could use that proton motive force to produce a lot of ATP. So the bacteria could grow higher in the water column where the oxygen concentration was higher and gain more energy from that. While the poor eukaryotes did not have that function, and so they had to remain low in the water column. But eventually one or more came together in that middle zone, and the started living together as mutualists, and eventually one of those bacteria that could do oxidative phosphorylation, an alpha proteobacterium, started growing inside those eukaryotic cells. Now the eukaryotic cells could start moving up in the water column and take advantage of niches that were previously unreachable to them. So now the mitochondria have many functions in those eukaryotic cells. They produce NADH in the TCA cycle through reactions starting from acetyl-CoA, which comes from pyruvate out of glycolysis in the host cell. Then they have 5 protein complexes involved in oxidative phosphorylation. The NADH CoQ removes a proton and electrons from the NADH. That is, they oxidize it. The electrons move through coenzyme Q, cytochrome reductase. Then cytochromes carry electrons to cytochrome C oxidase. Cytochrome oxidase combines electrons, oxygen, and protons to form water. That's complex 4 there. And at least 3 of those complexes move protons across the inner membrane from the mitochondrial matrix into the intermembrane space. And then those protons move through the ATPase, the ATP synthase, which is complex 5, to form ATP from ADP and inorganic phosphate. So many proteins are needed in this process, in addition to membranes, membrane proteins, ribosomal RNA, ribosomal proteins, tRNAs, DNA polymerase, RNA polymerase, etc. So a lot of genes are necessary. So in this diagram I've summarized some of what we know about how many genes are involved to make those proteins in each case. So here, right here, this is a mitochondrion with the outer membrane, inner membrane with the cristae, and the inner membrane space. And so the first thing you need is a lot of transport proteins to get those proteins that are expressed from the nucleus into the mitochondrion, either into the mitochondrial matrix or into the intermembrane space. And so there are two locations, so you need transporters, you need chaperone proteins, and many other things to get those in. So that's at least 10 genes, and it's probably might even be 10 times that amount. Also over here you need transporters to carry fatty acids, to carry pyruvate, and to carry amino acids into the mitochondrion. Each one of these can feed into the TCA cycle, which requires more than 18 genes to produce each one of those proteins, and probably at least double that number. So you have these transporters and you have the TCA cycle here, so count those up. You're probably up more than 50 additional genes involved in this process. And then you get to oxidative phosphorylation here. So you need about 72 genes make up complex 1, and that's the complex that oxidizes NADH to NAD+, and transport proteins across. And then there's complex 2, which takes at least 12 genes, probably at least double that, pumps more protons across. And then here's the succinate to fumarate part of the reaction in the TCA cycle. That's accomplished by complex 3, and it's also transferring electrons down the line. That takes at least 12 genes. And then complex 4, which takes oxygen, protons, and electrons to make water and gets energy out of that, and then pumps protons across. That takes at least 36 genes. And then the ATP synthase complex itself requires at least 8 to 12 genes, and so you have a number of genes there. Now, because DNA is involved, that is, the mitochondrion has its own genes, not very many, but it has about 50 genes. Then you need DNA polymerase to replicate that. You need repair enzymes in case there are any errors. You need RNA polymerase to transcribe the DNA into RNA. And you need ribosomal proteins to produce the proteins from those mRNAs. And that's illustrated over here. So just for replication, over 30 genes are needed to make the replication complex, and also repair enzymes. And about 100 genes or more are needed for the ribosomal proteins to produce the ribosomes, etc. About greater than 100 genes there, and for transcription, the polymerases, accessory proteins, etc., over 100 genes. So here, if you add them up, it's about more than 130 for oxidative phosphorylation, more than 18 for the TCA cycle, more than 30 for replication, 100 for transcription, 100 for translation, and membrane functions over 50. So that adds up to 438, and it's probably over 1,000, but minimally of what we know of those gene products, at least 438. So here's a representation of two mitochondrial genomes. The inner circle here is the human mitochondrial genome. It's about 16 to 17,000 base pairs, about the size of a typical virus. And then you have the yeast mitochondrial genome here, which is about 78 kilobases, much, much larger. About 1.5 lambda size, lambda phage genome. The blue areas here are the genes, so there's cytochrome B, there's a small ribosomal RNA subunit, large ribosomal subunit, one of the NADH enzyme genes there. The ones in red here are for tRNA, so quite a number of tRNAs, and then you have ribosomal RNAs. Well, of course, you would need those for the process of translation. So really very few genes compared to an alpha proteobacterium, which would, have approximately, 400 to 1,000 genes. Now we've just got about 30 genes, and most of those are tRNA genes and the ribosomal RNA genes. And then if you look at the mitochondrial genome of yeast, it also has a small number of genes, about the same number, but it also has introns. So there are areas that are sort of blank or yellow, and the blank areas contain no genes, or no genes that we know of. And the yellow areas are introns, so even these have introns. They're usually group 2 introns. So when you count all of these up, all mitochondrial genomes have between about 30 and 50 genes. There actually are a few that are smaller than that and a few that are a bit larger than that, with links from about 6 kilobases, 6,000 base pairs, up to 2,500 kilobases. And so these are getting to be more like bacterial size, but they only have 30 to 50 genes, very few genes. If you look at what types of genes those are, then there are ribosomal gene operons, that is a small and large subunit to make ribosomes. So you need that to do translation. And there are 20 to 30 tRNA genes, right? So you need that also to do translation. But as far as oxidative phosphorylation, where you need hundreds of genes, there are only 5 to 15 of those. And of the ATPase subunits, the ATP synthase subunits, only 2 to 3 out of the 8 to 12 that are necessary are present in the mitochondrial genomes of those mitochondria. So although over 400 to 500 genes are necessary, and maybe even more than that, to do all these functions, the mitochondria have only retained 30 to 50 of those. So as I showed you earlier in the video, then this has been happening where a lot of that DNA that was originally in the alpha proteobacterium has moved into the nucleus from where it's expressed. So you get transcription in the nucleus. Those messages move out into the cytosol, and then they're produced, they're translated into proteins. The proteins then are imported into the mitochondrion so it can perform the TCA cycle and oxidative phosphorylation and anything else that it needs to do. So this is why chloroplasm mitochondria have generally smaller genomes than do the bacteria from where they originated. So most mitochondria have genomes down in this range down here, but the alpha proteobacteria have DNA sizes that are up here in this area. So the mitochondrion, since being from being an alpha proteobacterium to eventually winding up as a mitochondrion, has lost more than 90% of its original genome. Okay, so that's the end of part 1 on organelles. I talked briefly about organelles in general, and then talked more specifically about mitochondria, both their origin and then what's happened to them since evolving as mitochondria from their original alpha proteobacteria ancestors. Okay, well, thank you. That's it for this video.